8. Parasite and host interactions. When considering the word parasite, various kinds of organisms may spring to mind, depending upon the audience. Veterinarians may think of scabies, mange mites, warble flies, helminths such as liver flukes, bird lice and disease organisms that cause rabies, plague, West Nile virus and bird flu. Medical entomologists may be more concerned about malaria, sleeping sickness and vectors of disease like mosquitoes and black flies. Agriculturalists would perhaps worry about plant pathogenic diseases and nematode parasites. Those involved with the biological control of weeds or insect herbivores may well think of diseases of plants and insects, and parasitoid wasps and flies. But springing to mind among only a few insect ecologists, perhaps, would be the insect herbivores themselves, caterpillars of the moths, butterflies and sawflies, larvae of chrysomelid beetles, bark beetles, weevils, grubs of root-feeding insects such as rootworms, and wood-boring larvae, such as cerambycid and buprestid beetles, and clearwing and carpenter moths. And yet, when the definition of the term parasite is appreciated, we can begin to recognize the vast convergence of many lineages toward the parasitic way of life. In common usage is the term host plant for insect herbivores, with the clear implication that plants are hosts to parasites. Explicit recognition of herbivorous insects as parasites is provided in the definition of a gall. Galls are abnormal growths formed from tissues of a plant or other host, due to the parasitic activity of another organism. Redfern and Shirley 2002, p. 207. This broad view of what is a parasite is somewhat controversial because of the ingrained emphasis on parasites of animals, particularly in the field of parasitology. However, plant pathology treats parasitic diseases extensively and we can observe life history convergence in parasites on animals and plants, section 8.6, so we should recognize the common features of all kinds of parasites. In this chapter we emphasize the diversity of insect parasites, as well as the convergence of many groups toward similar life cycles, their main characteristics and how they relate to host species. We compare parasites with other ways of life such as predation, emphasizing that intimacy and duration of relationships between parasite and host have major importance in understanding evolutionary pathways in parasite lineages, life history convergence, adaptive radiation and phylogenetic tracking of host lineages. Kinds of damage inflicted on hosts are discussed. Host responses in the form of defenses, how parasites can modify host behavior, and the population dynamics of host and parasite all contribute to a general appreciation of the parasite's ecological and evolutionary roles. 8.1 The parasite and parasitism. A parasite can be defined as an organism living in or on another living organism, obtaining its nutrition from that host organism and causing some degree of measurable damage to the host. We could add that parasites show some degree of adaptive modification to the host, but then every species shows adaptations to its food supply. Altogether, many of the species that are of concern to veterinarians, medical entomologists, agricultural and forest entomologists, and practitioners of biological control, are parasites. And those that study plant and herbivore interactions, parasite and host interactions, and multitrophic level interactions, in many cases are studying parasite-host relationships. Most caterpillars, but not all, spend their lives on a single host plant, eating plant tissues and causing real damage. They qualify as parasites. It is the durable interactions that characterize the parasite and its host, Combs 1995, 2001. This is true no matter what the systematic position of the partners, no matter the way the interaction is borne out, and no matter even that the benefits and costs of the association may be unequally shared. Combs 2001, p. 4. And so parasitism applies to the majority of insect herbivores. This view of the widely ranging groups of species that qualify as parasites is neither a trivial nor a semantic exercise. Recognition of broad patterns in nature is basic to the fields of ecology and evolutionary biology, so that the wider the comparative biology can be cast, the more general the hypotheses and theories can become to explain the parasitic phenomenon, the convergence of lifestyles, the tricks of the parasitic trade, the rates of speciation and adaptive radiation, and even perhaps the best methods of regulating their numbers. Reflecting upon the definition of a parasite, we should consider which organisms really qualify, and which do not. Parasitoids are organisms that have free-living adults that find insect hosts, on or in which they deposit eggs or living larvae. The larvae develop in a parasitic manner in the living host, whether the host continues to feed, or lie in a moribund state, paralyzed by the adult parasitoid. In either case, the host is killed eventually by the parasitic stage. Therefore, the adult parasitoid results in the death of many host insects, and its actions are equivalent to those of a predator. This was recognized by Reuter, 1913, who called them parasite-like predators, the parasitoidea, from which the word parasitoid is derived. Others have coined terms such as predatoid and carnivoroid. See Flanders 1973, illustrating a dichotomy of emphases. Although the facts are clear, parasitoid adults have impact on insect populations equivalent to predators, e.g., see Chapter 7, although their larvae are parasitic. And, as we shall see, the evolutionary biology of parasitoids is very much influenced by the constraints and opportunities afforded by the parasitic lifestyle. Those insects that may not qualify as parasites, strictly speaking, but which are considered in texts on medical entomology are the biting flies, mosquitoes, black flies, biting midges or no-see-ums and most adult fleas. Of course, they are important in medical entomology as vectors of serious diseases, but they enjoy only brief encounters with the mammalian, reptilian and avian individuals, and even other insects, from which they obtain a blood meal. Adults are free-living, as are the larvae. Were they true parasites, they would not be effective vectors of diseases at all, because a parasite would live on a single host and not move among hosts as vectors must do to be effective. Even though a flea may stay on one host for a while, it can easily hop from one host to another, and is more likely to do so than a small caterpillar feeding on a large tree. However, here we begin to recognize the gradient between parasites and free-living organisms, for as in most of biology, humans' classifications fall short of capturing the reality of nature. See also Lafferty and Curry's 2002. We should also recognize that, as a way of life, parasites are involved with much of ecology, both basic and applied. They are ubiquitous and exert impacts on almost all living organisms. Parasites dominate food web links. Lafferty et al. 2006, page 11211, and Parasitism is the most common consumer strategy among organisms. Lafferty et al. 2008, page .533. They are major contributors to malaise and mortality with often strong impact on host survival, and the distribution, abundance and population dynamics of many species. Parasites play a major role in ecology and evolution. Hence, we should understand them well, recognize common features observed in this lifestyle, and explore the evolutionary potential of these kinds of organisms that are easily overlooked in nature, and often ignored by ecologists. 8.2 Kinds of parasites. While the definition of a parasite is straightforward enough, the range of interactions that are considered in the literature as parasitic is enormous. Some kinds of interactions and their definitions contribute to an appreciation of the scope of parasitism and the considerable variation in the intimacy between host and parasite. Table 8.1. Even the lexicon relating to the parasitoid life cycle has become extensive because of the diversity of interactions involved. Table 8.1. The recognition that many insect herbivores act as parasites dates back over 30 years. Price 1975c, with Bush, 1975, considering speciation in Phytophagous parasitic insects, and Janssen, 1975, noting that bruchid weevils act as parasitoids, with the adults finding seed pods and seeds on which they oviposit, while larvae feed slowly in the seeds as parasites, eventually killing them. Just as in our chapter on parasites, Janssen noted that when considering bruchid diversity, it will be most profitable to think of them as parasitoids. Page 157, noting their impressive host plant specificity, with 83% of species in his sample known from only one host plant species. Plant pathogens similarly show narrow host ranges. Van den Akerfken and Bonas, 1997. It was clear then, as it is now, that intimate and durable relationships between host and parasite necessitates specificity in feeding, fosters isolation between populations and form the engine of adaptive radiation in plant and animal parasites alike. The importance of finding broad similarity across taxa in the durability and intimacy of interactions, which characterize the parasitic relationship with hosts, lies in the discovery of broad patterns in nature. The broader the pattern, the more general the concepts can become, and the wider ranging the theory. When we can recognize massive convergence in life history traits, host relationships and adaptive radiations, across several trophic levels, we have achieved a major conceptual breakthrough. General characteristics of parasites are considered next, including concepts treated earlier by Price, 1977, 1980. 8.3 The number of parasitic insect species. Various estimates have been made of the numbers of parasitic species in any fauna, and their proportional representation in that fauna, ranging from about 15 to 50%, e.g., Arndt 1940, Rothschild and Clay 1952, Askew 1971, Bush et al. 2001. We can make our own estimate using 1,320,000 and growing estimated described animal species in the world. Hawksworth and Kalin Arroyo 1995, 105,170 metazoan parasites. We can make our own estimate using 1,320,000 and growing estimated described animal species in the world. Hawksworth and Kalin Arroyo 1995, 105,170 metazoan parasites on animals. Poulan 2007 plus 82,131 parasitoid species. Godfrey 1994, a total of 187,301 Increase the percentage, not to mention the undescribed species concentrated in the bacteria, protozoa, viruses, nematodes, fungi and insects, all with many parasitic species. But if we concentrate on the insect fauna, which after all dominates in numbers of species in any locality, some surprises may be evident. The British insect fauna is well studied and documented, with well-developed species lists. Cloet and Hinks 1945, 1964-1978, which can be employed in estimating the relative abundance of various feeding types. Table 8.2. Most notable in this compilation is the large proportion of the fauna represented by insects with a parasitic lifestyle, a total of 70.7% of the fauna. Also, in every case in which both predators and parasites are represented in an order of insects, parasites have become much richer in species than the predators. The most remarkable example of species richness is the parasitic Hymenoptera, the parasitoids, with 5,342 species, representing 89% of the parasites on animals and 26% of the entire British insect fauna. Even in the updated edition of the British Hymenoptera, Fitton et al., 1978, page B, it was stated that the total will be considerably increased when the parasitic are better known. When we dissect the richness of parasitic Hymenoptera into families, we also observe that many families rank among the richest in the British fauna. Table 8.3. While the 10 largest families of predatory insects average 45 species per family, herbivorous parasites have a mean of 329 species per family and carnivorous parasites reach a mean of 489 species per family. Among the carnivorous parasites, most families are composed of parasitoids, and most are hymenopterans. The Tachinidae with 228 species is a large family of dipteran parasitoids. The Philopteridae Pyraptera, with 176 species represented, is the family of bird lice, the only non-parasitoid family listed. 8 of the 10 largest families of carnivorous parasites are hymenopteran parasitoids. Notable also in Table 8.3 is the large size of many herbivorous parasitic families. The Cystidomyidae are the gall midges, with an intimate host plant and parasitic interaction among the species. The weevils Coleoptera, Curculionidae are mostly internal parasites of plants as larvae, burrowing in stems, in cambium, under bark and in roots, shoots and fruits. Other families include lepidopterans, hemipterans, sawflies and beetles, so that all the major orders of insects are represented. In rigorous phylogenetic tests of the extent of adaptive radiation of non-phytophagous versus phytophagous insects using 13 sister group comparisons, the phytophagous groups were larger in 11 cases or 85% of cases, Mitter et al. 1988, see also Mitter et al. 1991, Farrell and Mitter 1993 and Chapter 4. However, at the higher trophic level, Wiegmann et al. 1993 found that in 9 of 15 comparisons the carnivorous parasitic taxon was smaller than Thenan parasitic group, see also Winkler and Mitter 2007. That more local and intensive studies once British fauna should provide a view different from a worldview based on much less detailed studies hold not be surprising. Also, detailed research on a tropical fauna using molecular techniques to identify cryptic species is revealing much greater parasitoid species richness and much higher specificity than previously recognized, Smith et al. 2006, 2007, 2008, Janssen et al. 2009. The high numbers of species in families that are largely represented by parasitic lifestyles raises the question of why such extensive speciation should be apparent, relative to the smaller families of predatory insects. The answers are discussed in the next two sections. 8.4 Small body size. Obviously, parasites are smaller than their hosts, and usually much smaller. Living in or on the host means that the host not only provides food to the parasite, but also a habitat in which to live. Considering a large plant, like an oak tree, or a large mammal, such as a human, we can begin to appreciate the richness of habitats available on a single host. The mammalian integument is one example of the myriad places in which parasites can live. Surface temperatures and humidity vary greatly over the body. Pelage can be dense or sparse. Sites may be easily groomed or protected. Cavities differ. Glands differ in shape and secretions, and hairs, and their follicles, vary in size and shape. The landscape of the mammalian body, such as the human, becomes as varied and topographically complex for a parasitic mite as the state of Arizona may be for a human being. Figure 8.1. When considering parasites and their small size, as Thompson, 1942, page 77, wrote, we have come to the edge of a world of which we have no experience, and where all preconceptions must be recast. We are prisoners of the perceptions of our size, and rarely recognize how different the world must appear to small animals. Gould, 1974, p. 21. We seldom think of our own bodies as ecosystems on which a myriad of species live, and it would take a dermatologist or a microbiologist to fully appreciate the complexity of the body surface, and the ways in which species become fine-tuned and adapted to only small and specific locations on the skin, e.g., Marples, 1965. The implications of small size are almost endless. Body design, physiology, life cycle, population size and dynamics, niche occupations, community structure, evolutionary potential and adaptive radiation are all inextricably involved with size of the organism, e.g., Thompson 1942, Bonner 1965, Peters 1983, Calder 1984, Andrews 1991. Ecologically, it is easy to imagine that parasites can live in tiny niches, many species can coexist on a host, but in discrete places, and the amount of food consumed during a lifetime is minuscule. Also, small distances may result in sufficiently large divergence in the environmental quality that habitable patches may be narrowly defined, with disjunct distributions to be expected, and reproductive isolation occurring over short distances. Short generation times, and high populations, coupled with life on hosts affording discrete microhabitats, may well provide the ingredients for rapid speciation and adaptive radiation. Entering into the lives of parasites provides many challenges, but after all, as ecologists we should try to understand these species that in numbers may well exceed all other kinds of organisms. This perspective and the effects of body size will be discussed many times in this chapter and elsewhere in this book. Table 8.5 Adaptive radiation of parasites. Many reasons exist for the extensive adaptive radiation of parasitic groups, but high specificity to host species appears to be the most salient feature. Parasites are usually highly specific, often adapted to one species of host. This is the general case among parasitic groups on plants, such as leaf mining flies, Agromyzidae, plant bugs, Miridae, and leaf mining, leaf rolling and tying, and gall-inducing lepidopterans in the family Gulecchiidae, Table 8.4. It is also a feature common to animal parasites, with parasitoids showing that 53% of ichneumonids and 60% of braconids are recorded from a single host. Molecular studies are showing even higher levels of specificity, with over 90% of parasitoid species attacking only one or two host species, Smith et al. 2008. Bird lice are highly specific also, with 87% of species recorded as specific to a single host species. These levels of specificity contrast strongly with predators of seeds and insects, and plant grazers, which typically forage on 10 to 30 species of food items, Table 8.4. Seldom have arthropod predators been evaluated adequately for diet breadth. One well-studied scorpion, Paruroctonus mesensis, is recorded to consume more than 125 prey species including a small snake, McCormick and Polis 1990, Polis and Yamashita 1991. So far, there has been a direct relationship between the duration of the study and the number of species determined to be in the diet, McCormick and Polis 1990, p. 302. Thus, the listings for vespid wasps, spiders and scorpions grossly underrepresent the variety of prey. This is because prey are often recorded by higher taxa, such as orders and above. We begin to see the reasons for high specificity among parasites when the higher and lower ends of the ranges in specialization are examined. Those families with higher than 50% of species with only one host have a very intimate relationship with that host, with individuals living in or on that host for the majority of their life cycles, or all of it in the case of the wingless philopterid bird lice. In contrast, phytophagous myrid bugs are good walkers, even as nymphs, and can move around from plant to plant, encountering several potential host species in a lifetime. The same is true for louse and bat flies, Hippobosidae, which are parasitic on birds, bats and other mammals. Many species are winged as adults, and pupariate in the soil, so that a disconnection exists between host and parasite, and the freely foraging adult may find opportunistically various hosts before it settles and sheds its wings. Examining the specificity in families of parasitic insects on mammals and birds supports the hypothesis that intimacy of life cycle between parasite and host selects for higher specificity. Philopterid lice are wingless, with a life spent entirely on the host, with 87% of species recorded from a single host, table 8.5. Without wings, opportunities for parasite transfer to other hosts arise mainly among conspecific individuals, parent to young in mammalian skin habitats. Topologic sites of prostigmatic symbiotes E. M. N. S. O. P. T. Particular, and among nestlings. The opportunities for host species shifts are very limited, resulting in close tracking of host lineages and parasite lineages. The ectoparasitic flies on bats, Streblidae, have blood-sucking, winged adults which deposit fully developed larvae on the surface of bat roosts where the larvae pupariate. So adults emerge close to the original host species and are likely to settle on such hosts, maintaining an intimate relationship with that species and even that particular colony. However, other bat species in the roost may be exploited, reducing specificity in this group relative to the bird lice. The bot and warble flies, Estridae, show less specificity than Streblids, presumably because the adults are strong flyers and move freely to find mammalian hosts. However, the parasitic life of the larva still requires special adaptations for living inside the host, and almost 50% of species are recorded from a single host species. Some of this specialization is determined by the remoteness of the hosts, such as reindeer in the northern tundra, and some strange noses, such as the elephants, hippopotamuses and the Histratopsilidae is a family of fleas that utilize mostly small mammals as hosts. Adults take blood from hosts, but can jump from host to host, and are long-lived. Larvae are free-living, usually feeding in nest debris. This lifestyle results in relatively loose relationships with hosts, and relaxed specificity, with some species reported from over 20 mammal species. The least host-specific group in this progression is the Hippobosidae, or louse flies, with the main hosts being birds. The louse flies are related to the Streblids, but intimacy with the host is lost when the mature larva drops to the ground to pupariate in the soil. Hence, an emerging adult must fly around in search of hosts and may encounter many species, from which it may gain a blood meal, and so specificity is reduced. The examples in Table 8.5 show a triangular pattern of host specificity, with high specificity on the left, and no parasitic species attacking more than 5 host species, and low specificity on the right, with records showing species from over 80 host species. Where there is high host specificity, we should anticipate that reproductive isolation becomes stronger when host shifting occurs, and speciation may become more extensive. The number of hosts available in the lineage of hosts is also probably an important contributor to the potential for adaptive radiation of parasites. Host species probably act as ecological islands, just as islands in oceanic archipelagos act to promote geographic and reproductive isolation in the celebrated cases of adaptive radiation. When we compare numbers of recorded hosts, and parasite specificity, and the number of parasite species per family, a suggestion of such a trend is evident. For example, the philopterid lice on birds exploit about 8,600 species of hosts. They are highly specific parasites and exhibit extensive adaptive radiation with about 1,500 species worldwide. In contrast, with fewer hosts and/or lower specificity, adaptive radiation in the parasites declines into the hundreds of species. Price 1980. Similar scenarios can be developed for insect herbivore groups, e.g., Price 2002a, as suggested by the richness of species listed in Table 8.3. For example, the gall midges, Cicidamiidae, are highly specific to one or a few host plant species, as is the case in most gall-inducing insects. The extent of adaptive radiation is very high because of this specificity, and the richness of host plant species in the lineage or lineages exploited. This has been observed within the family Societomyidae, Roscom 1985, figure 8.2, in other families of galling insects, Price 2005, and in other kinds of arthropod parasites, Price 1980. Altogether, data support Eichler's rule, Eichler 1948, developed for animal parasites, which states that the diversity of the parasitic fauna will increase as the diversity of the host taxon increases. 8.6 Life History Convergence. A problem common to many parasites, with their small size and widely scattered hosts, is the transmission from one host to another, or the infection or colonization of new hosts. Overcoming this problem has frequently involved a complex life cycle with the massive generation of individuals of a particular stage adapted for transmission and infection of new hosts. In many cases, complex life cycles have evolved with several distinct stages, some of which involve asexual reproduction and one stage with sexual reproduction. This convergence is observed in flatworms like liver flukes. Platyhelminths, Digenea, gall aphids, gall adelgids and rust fungi, figure 8.3, representing 3 taxa of widely different origins, which utilize vertebrates or plants as hosts. Frequently, more than one host animal or plant species is used in the life cycle, as in the cases shown in figure 8.3, but also where insect vectors become the transmission agent for microbial organisms causing diseases such as malaria, yellow fever, Chagas disease, sleeping sickness and bubonic plague, Bush et al. 2001. Many plant diseases are also vectored among host plants by insect herbivores, particularly sucking insects such as aphids, leafhoppers, planthoppers and whiteflies, but also bark beetles. Plant diseases include mosaics, wilts and blights caused by viruses, mycoplasmas, spiroplasmason bacteria, and many multiply in the vector, causing disease in the vector, but enriching saliva with parasite stages ready for injection into the next host. Maramorisk and Harris 1979, Borer et al. 1992, Nault 1994, Malloy and Murray 2001. The high production of propagules for transmission to a new host among disease organisms no doubt causes serious problems for vectors and recipient vertebrate hosts as well. Among humans, for example, high mortality results from the massive reproduction of such propagules, which result to ensure that tiny doses of blood taken up by insect vectors contain at least a few infective stages necessary to continue the parasite's life cycle. Ewald 1983, 1994. Of course, many insect and parasites are well equipped to move from host to host, because adults are winged and commonly fly efficiently. But they, too, show reproductive capacities consonant with the problems faced in finding hosts, and surviving in hosts. For example, some parasitoids oviposit onto foliage. An infection of a host occurs when an egg is eaten or an active larva hatching from an egg attaches to a passing host. In these cases parasitoid fecundity is very high, compensating for the low chances of host infection, figure 8.4. At the other extreme are parasitoids which oviposit directly into the host stage in which the life cycle is completed. Fecundity is low in these parasitoids because the probability of survival is high. Again we see convergence in reproductive strategies, this time in parasitoid taxa, such as the Hymenopteran ichneumonids and the dipterantachnids. As with all adaptive strategies, there are associated inevitable limitations or constraints. When such strategies are broad-scale in general, as in the parasitic lifestyle, we observe repeatedly the convergence of traits under similar kinds of natural selection. Parasitoids are constrained by the survivorship curve of the host, as if in a straitjacket that limits life history options and selects for convergence, see figure 8.4. Given the richness of parasite taxa, the opportunities for comparative biologists are immense for the discovery of general patterns in nature. 8.7 Convergence among parasitoids. We concentrate here on parasitoids because of their immense interest to ecologists, their role in pest population regulation, and because we need to understand with clarity how different parasitoids are from predators when we consider the parasitic way of life. Evolutionary convergence into the parasitoid way of life provides one of the best and most diverse examples available today, convergence being the paths of disparate taxa evolving towards a common body plan and/or life history type. We see similar lifestyles evolving even from across orders of insects. Also, within each lineage the adaptive radiation onto new hosts, and into new ecological niches, is extreme in some cases, as, for example, the ichneumonidae with an estimated 15,000 described species and perhaps 60,000 total species in the world fauna. Godfrey, 1994, lists the families of insects which contain parasitoids, including 37 families in the Hymenoptera, 9 families in the Diptera, 5 in the Coleoptera, 2 in Lepidoptera and 1 in the Neuroptera. Families with 1,500 or more named parasitoid species in the world include the Chalcididae 1,500, Tauromyidae 1,500, Pteromalidae 3,100, Incertidae 3,000, Euliphidae 3,000, Diapridae 2028, Scelionidae 2,768. Ichneumonidae 15,000, Braconidae 10,000, Typhiidae 1,500, Bombylidae 3,000, Tachinidae. Egg Fundatrix gallicola sissons progrediens 8,200 and Meloidae 2,000. A total estimated parasitoid fauna in the major insect orders add up to well over 50,000 species in the Hymenoptera, about 15,000 species in the Diptera, and over 3,000 species in the Coleoptera. There is hardly an insect family that does not provide hosts for parasitoids, and many other invertebrates are also parasitized. Parasitoids outnumber all chordates, fishes, amphibians, reptiles, birds and mammals. With such vast numbers of species we should not be surprised at the impressive diversity in modes of reproduction and life history. Many specializations are not observed in solitary predators: polyembryony, precocious larva and caste formation, haplodiploidy and sex ratio allocation, parthenogenesis, heteronomy, inbreeding or sib mating, hypermetamorphosis and extended phenotypes. These adaptive traits will be described briefly and more details can be found in other sources, e.g., Charnov 1982, Godfrey 1994, Quick 1997, Strand and Gerbic 1997, Combs 2001. The examples illustrate, apparently, how favorable the parasitic habit is to the evolution of novelty, as also seen in morphological innovations. Such creativity argues against the views of earlier biologists who suggested that parasites as a whole are worthy examples of the inexorable march of evolution into blind alleys. Noble and Noble 1976, page 525. Most specialization leads to blind alleys. Meyer 1963, page 596, and specialization is a deepening rut in evolution. Huffaker 1964, p. 645. Polyembryony, the formation of multiple embryos from one egg. This condition is known in 4 families of parasitoid wasps: Incertidae, Platygasteridae, Dryinidae and Braconidae. Strand and Gerbic 1997. Brood sizes can reach up to 3,000 wasp progeny per host, with clear impact on population growth. Precocious larva and caste formation. Polyembryonic incertids produce polymorphic larvae, with one type sterile and precocious. They develop rapidly and act as a soldier caste, which manipulates sex ratio of the reproductive caste and protects the brood from other parasitoids attacking the host caterpillar. Haplodiploidy and sex ratio allocation. Sex determination in which males result from unfertilized eggs and females are produced from fertilized eggs. Since the ovipositing female can manipulate sex ratio, various adaptive traits appear. Males can be produced on poor quality hosts, perhaps small hosts, and females can be placed on higher quality hosts. Sex ratios can be strongly skewed in favor of females, thereby increasing population growth. Parthenogenesis. Haplodiploidy is a form of parthenogenesis or renotoky, but in addition, the Litocus parthenogenesis, in which only females are produced from unfertilized eggs, is moderately common in parasitoid wasps, reported in 20 families and 205 species. Heteronomy. Parasitoid species in which the two sexes develop in different host types obligatorily. This trait is observed in aphelinid parasitoids in which females develop as an endoparasitoid of homopterans such as mealybugs, whitefly and scale insects, while males develop as hyperparasitoids, utilizing females of their own species or other species. Inbreeding or sib mating. Where sex ratios are strongly female biased among gregarious parasitoids, a few males from one host will mate with their sisters originating from the same host. Hypermetamorphosis. The life history in which larval instars develop through more than one form, as in blister beetles meloidae, with an active long-legged first instar larva, the triungulin, which can search for host grasshopper eggs or bees' nests. They have less active parasitic stages and legless later instars. Also, first instar larvae of many endoparasitic species are equipped for fighting and killing conspecific and heterospecific endoparasitoids. Large, rigid head capsules with strong mandibles result in vicious weapons of attack. They usually molt into second instars with smaller mandibles less equipped for physical aggression. Planodial first instar larvae of parasitoids, as described below, are equivalent to triungulins. Pinto 2003. Planodial larvae are found, for example, in the Eukaryotidae which utilize ant pupae as hosts, Paralampidae, see below, Euceros, see below, and acrocerid flies. Extended phenotypes. Parasites can affect their own environments such that the phenotype of the parasite extends beyond the organism itself, such as a gall-inducing insect, a parasite, stimulating gall formation on the host plant. Dawkins 1982. Combs 2001 has emphasized that the durable interactions of host and parasite mean that genomes of both interact for much of the life of the parasite. The host becomes the extended phenotype of the parasite, and the parasite becomes the extended phenotype of the host. The holometabolous insect host offers a series of substantially different resources which can be attacked by parasitoids, each requiring different specializations which ensure success. As an example, a sawfly such as the Swain jack pine sawfly, Neodyprion swaini, passes through 4 stages: egg, larva, pupa and adult. Figure 8.5. In addition, larvae pass through several instars as they grow, ending with a non-feeding eonymph, which enters the forest litter and spins a cocoon in which it overwinters, pupating in the spring and emerging as an adult. Parasitoids attack eggs, larvae and eonymphs, and larvae, pupae and new adults in cocoons, but not emerged adult sawflies. Many kinds of life history are illustrated by the Neodyprion parasitoids. Price and Tripp 1972. 2 species of egg parasitoid attack and emerge from the egg. Larval, eonymphal and cocoon parasitoids are primary parasitoids and emerge from cocoons in the spring. Paralampus Hymenoptera. Paralampidae oviposits on foliage, active first instar larvae and planidia, hatch and wait for a passing sawfly larva, onto which they climb, bore into the host and become endoparasitoids. Spathymygoniea diptera. Tachinidae is a larviparous species, which injects first instar maggots into feeding sawfly hosts, where they become endoparasitoids. Olesicamp Hymenoptera. Ichneumonidae attacks early instar larvae, and Lamachus, in the same family, attacks later instar larvae. All larval parasitoids are koinobionts because they do not interrupt feeding and development of host larvae, but finally kill the host in the spring before it can pupate. Exenterus, another ichneummonid, is a specialized eonymphal parasitoid, laying eggs in the cuticle of the host. Secondary carnivores. Euceros. Paralampus. Phygaeduon Paralampus Pleolophus Mastris Jellies Dalbominus. Primary carnivores. Herbivore. Before the sawfly leaves the tree and spins a cocoon, The parasitoid larva hatches onto the surface of the host after a cocoon has been spun and feeds as an ectoparasitoid in a coinobiont. The cocoon parasitoids are mostly ichneumonids, Pleolophus, Endices, Mastris, jellies and agrothyroids. All pierce the cocoon with long ovipositors, inject the softfly host with venom, thereby paralyzing it, and oviposit on the host surface. Larvae feed ectoparasitically as idiobionts.